phospholipids. Dipesh Tiwangan, roll number 31. Devpriyank Sharma, roll number 32. Devika Diwan, roll number 33. Dhruv Patel, roll number 34. Dinesh Bairwal, roll number 35. Myself, Divija Singh, roll number 36. Domendra Kumar Sahu, roll number 37. And Isha Minch, roll number 38. So further, we are going to know how glycogen is produced from a simple molecule which is glucose. And this process is called glycogenesis. This is glucose, which is a simple sugar. It is one of the most vital biomolecules for generation of energy in the form of ATP. Now, we are going to see how glucose will be acted upon by an enzyme hexokinase.
छन से जो टूटे कोई सपना, जग सुना सुना लागे, जग सुना सुना लागे, कोई रहे ना जब अपना जग।
This reaction cannot occur in the absence of antinucleus, which is the main cofactor for the enzyme hexokinase.
बम बम बोले मस्ती में डोले, बम बम बोले मस्ती में तू डोल रे, बम बम बोले मस्ती में डोले, बम बम बोले मस्ती में तू डोल रे।
Glucose has been phosphorylated to glucose 6-phosphate by utilization of one molecule of ATP. Further, glucose 6-phosphate is transformed into glucose 1-phosphate by the enzyme nuclease. Next, more glucose units are added to the previously formed complex by the same enzyme. Further, glucose units are added in the presence of two enzymes, branching enzyme and glycogen synthase, to form the final product, which is glycogen.
दीवार टूटती क्यों नहीं है?
टूटेगी कैसे? अंबुजा सीमेंट से जो बनी है। अंबुजा सीमेंट।
Since some monomer units are left in the branch chain, they are transferred to the main chain by the enzyme glucan transferase. Glycogen phosphorylase cannot break the alpha-1,6 branch bond. For this action, debranching enzyme is required.
You make me a believer, believer. You make me down, you build me a believer, believer.
The debranching enzyme will break the alpha-1,6 branch bond and remove the last monomer unit. Further, the main polymeric chain is spliced by the enzyme glycogen phosphorylase and breaks into glucose 1-phosphate molecules. Next, glucose 1-phosphate is converted into glucose 6-phosphate by the enzyme phosphoglucomutase. Glucose 6-phosphate is dephosphorylated to form glucose by the action of enzyme glucose 6-phosphatase. Hence, glycogen is broken down into glucose, its monomer. This process takes place in the cells of muscle and liver tissues in response to hormonal and neural signals. In particular, it plays an important role in flight or fight response and the regulation of glucose levels in the body. Now, we will try to understand how glycogen metabolism is regulated. Two hormones regulate the glycogenolysis and glycogenesis, which are epinephrine and glucagon. Firstly, the hormones bind to their specific receptors present on the cells of liver and muscle. Then, the hormone receptor complex found in turn helps in the conversion of ATP to cyclic AMP with adenine cyclase. Then, cyclic AMP is formed which activates the inactive protein kinase to the active protein kinase. Now, the active protein kinase complex is formed which will inactivate glycogen synthesis by inactivating the enzyme glycogen synthase. This enzyme is inactivated by phosphorylation, which in turn inhibits glycogen synthesis. Active protein kinase form also helps in conversion of inactive phosphorylase kinase to active phosphorylase kinase by phosphorylation and utilization of one molecule of ATP. This activates phosphorylase kinase, which in turn favors the glycogen breakdown by conversion of inactive glycogen phosphorylase to the active glycogen phosphorylase by utilization of one molecule of ATP again. Both the hormones promote the formation of glucose. Next, we will see allosteric regulation of glycogen metabolism. The level of glucose 6-phosphate regulates the process of glycogenolysis and glycogenesis. So, glycogen synthase is allosterically inhibited by glucose 6-phosphate. It is allosterically activated by glucose 6-phosphate, and glycogen phosphorylase is allosterically inhibited by glucose 6-phosphate. Hence, the key enzyme for glycogenolysis is phosphorylase, which is activated by glucagon and epinephrine. The key enzyme for glycogen synthesis is glycogen synthase, with the activity of which is decreased by epinephrine. The process of glycogen synthesis and glycogenolysis has many steps with many enzymes. So, if any enzyme is deficient, the process is affected and deficiency manifestations are seen. Some of these diseases are listed here. So this was our role play on glycogen metabolism. Thank you.
Is my name, это мой money rain, no pain, no gain, иди нахуй Monday. Дора is my name, ра-та-та, бэнг-бэнг, заряжаю, бля,